Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and welcome back. Welcome back, and this session is going to be very special because we are going to start our new journey today. Good evening all of you. Good evening all of you guys. How are you all? Yes guys. So please let me know if the video is video is absolutely normal. Oh. All right, guys. Quickly let me know. Everything is audible, visible. Good evening, guys. Good evening. Welcome back. Net. This class, yes, good evening, guys. This class is only on YouTube. This class is special because this class is the first live demo session of the classes which will be held in your app. So quickly, let me know if all is good and we can go ahead with the session. Yes, please. It's lagging a little. Let me check. Karo. Yes, guys. Now it will be absolutely fine. Yes, please. Right. I'm fine now. Phone. All right, guys. All right. So, guys, this is Phoenix 2.0, and this is our first class of the new batch that we are launching for NEET 2027 preparation. And guys, what is so special about Phoenix 2.0? Let me give you a little idea about that. I'm sure you already know about this, but guys, this batch is for NEET UG 2027 for all the class 12 and dropper students. And what will happen in this Phoenix batch, guys? This Phoenix batch is a collective effort of you and us, all my team members, to help you crack NEET with flying colors. So this is our dream. So we are going to work on this dream together in this batch. So guys, if Sunday, that is 10th May, this is the last day for enrolling in the batch with this price fees. So if you use this code, you can get this batch for just 4,699, guys. And what is so special, guys, if you take this batch, you get 600 plus live lectures. Not only you can watch my lectures, you can also watch any educator's lectures. You can choose your own educator, right? Good evening, guys. New student, all clear? And this is only till 10th May, guys. After 10th May, that is after tonight, the fees is going to be revised. And why should you hurry? Because the batch is going to start on 20th May. This session, guys, is particularly to give you an idea what happens in our batches, and you have to enroll quickly. So if you, as already said, if you take this batch, then you are open to watch the lectures of all other educators also. Now I want to quickly start with the first lecture, first chapter of class 12, which is sexual reproduction in angiosperms. Now I know when you talk about this chapter, there are certain terms like microsporogenesis, megasporogenesis, microgametogenesis, megagametogenesis, then microspil, microsporangia, and so on. These terms are confusing. So in this lecture and in this whole batch, guys, I'm going to make things crystal clear for you. Let's start with the first chapter and see how we are going to go about it. Now see, the chapter is sexual reproduction in flowering plants. Quickly give me a thumbs up, send me hearts that you're all ready. And please sit with your copy and pen. Keep your books aside. We are going to understand the things in detail. Conceptually right, we have to be. 11 portions, yes, Dharuna. 11 portions will also be covered because these batches are for 12th and droppers. So first, So it is going to be a mixed match, right? So we'll do class 12 as well as class 11 together. So after class 12, we'll be doing some class 11 topics. So it's going to be a wholesome preparation. You're a part of my batch already? Oh wow, Purvish. All right, guys. So you're ready with your copies and pen? Can we start with it? Can we start with it? Quickly share this session with your friends, guys. Quickly share it with your friends so that we can go ahead. Give out the syllabus flow. Okay, Purvish, the whole syllabus will be shared with you on the Telegram channel also. This session is about the live demo of how things are going to run in our batches, right? So how and why you should take these batches. So otherwise, every day is marked. Every day is marked in the next whole preparation, in our preparation. So things are going to be very, very organized. You will be getting the proper notes. You'll be getting DPP based on what we have studied in the class. And every chapter is divided into certain lectures. And it is a day-wise preparation. We are going to have class every day because I'll be taking care of both botany and zoology for NEET 2027, right? So our classes will be daily. All right, perfect. Now let's start with the session. I'm very excited. Now see, sexual reproduction in flowering plants, guys. So there are certain terms that you must understand. First of all, the term is sexual reproduction. So when we talk about sexual reproduction, be it plants or be it animals, the sexual reproduction consists of certain events. There are certain events which occur in sexual reproduction. Now, what are those events? Let's see. What are those events? Number one, there is always gamete formation. Sexual reproduction is characterized by gametogenesis, that is gamete formation. So there will be male gamete formation and female gamete formation. How and where and when, we are going to discuss it one by one. But the events that are going to take place, first is gametogenesis. Second, the gametes must come together. So there has to be transfer of gametes, and if we talk about plants, then this transfer of gametes occur by the process known as pollination. It occurs by the process known as pollination. Now, once they come together, once they are transferred and they are in vicinity, they come together, what will happen? The only thing that can happen is fusion of the gametes, which is known as fertilization. That is fertilization. Then followed by fertilization is zygote formation, and zygote formation will be followed by embryo formation, right? Embryo formation, and we are talking about plants, so this will result in the formation of seed. So these are the events that are going to take place. Please let me know that all these points are clear. All these points are clear to you, right? Easy. These are the things that we have already studied since we have ever started talking about flower, right? Now see, another part of this chapter's name is flowering plants. We are going to talk about angiosperms. We are only going to talk about sexual reproduction in angiosperms. Now, when the word angiosperm comes in, the difference and the characteristic things that are going to happen here is that fertilization, which occurs in other plants also and other animals also, unlike that, in guys angiosperms, it is always a double fertilization. It is always double fertilization. And then fertilization is not only followed by the zygote formation, it is also followed by the formation of pen. Now, what are these things? We'll be discussing it. And embryo formation is also going to occur along the endosperm formation. And then what is a seed? We are going to discuss everything in detail. Is that clear? So these are the events which will take place. And obviously, guys, we can say that the first two processes, that is gametogenesis and pollination, these two processes they comprise pre-fertilization events. They can be known as pre-fertilization events, right? This being the fertilization, and the events like zygote formation and pen, embryo formation, endosperm, and seed formation, these are called the post-fertilization events. These are called the post means which happens after This means before, post means after, post-fertilization events. Are these points clear? Now, since we are talking about the flowering plants, guys, let me make this thing very clear right here that flower is a special structure. It is a specialized structure meant for sexual reproduction. There is no other motive of a flower. So, when we are talking about sexual reproduction, where does it occur? It occurs in flower. It occurs in flower. It occurs in flower, and that is why, guys, we call flower as a reproductive unit. We call flower as a reproductive unit. Why some flowers are so attractive while others are not, why some are so bright and showy while others are small and inconspicuous, we are going to find reasons for all this in this chapter. Is this point clear? Is this point clear to all? Please let me know. Yes, guys, please let me know. All clear? All right. Now, occurs in the flower, which is the reproductive unit. Now, let's first discuss more about flower. Let's first discuss more about flower, and then we will see the process. Now, if we talk about flower, guys, flower, as I've just mentioned, it is a reproductive unit. It is a reproductive unit. Clear? Also, flower, as NCERT says, is a modified shoot. Is a modified shoot. It is a condensed shoot. Okay, we'll come on to the condensed part in a while. Now, it is a modified shoot. Clear? If we have some understanding of plant kingdom, then we must know, yes, guys, then we must know that in the previous stages of plant kingdom, that is still gymnosperms, there were some special structures called sporophylls. Remember that? Sporophylls were those units, those specialized leaves which bear spores. Now, what happens in a flower? So, flower is basically a bunch of some similar sporophylls, but how is it different? See, first of all, why did I call it a modified shoot? So, there is this theory which says that basically the shoot has condensed, shoot has modified into a flower, where the leaves have modified themselves into the floral appendages. And since leaves arise on the nodes, and similarly leaves, they arise on nodes, so does the floral appendages. So does the floral appendages on the nodes. The difference is, now here when it becomes a flower, these nodes become closer, nodes come closer. That means internodal condensation occurs. Internodal condensation takes place. Internodal condensation takes place, and all these nodes come to lie on the, yes, they come to lie on the thalamus. Now, how, sorry. So, see, let's say if this is a stem, and these are the nodes, as we know, these are the nodes, and at every node is a leaf. Now, instead of a leaf, what happens? First of all, please let me know. Post-fertilization events are not mentioned. Where are they not mentioned, darling? Where are they not mentioned? We have just read about the post-fertilization events. These are post-fertilization events. They're all in your syllabus, and we'll be understanding them one by one. We'll be understanding them one by one. Is that clear? Yes, please. Okay. Now, moving on to the details of the flower. Please understand, how is a flower formed? So first of all, everything that has to appear on the plant arises from the node, because node contains what? Node bears the meristem, right? Now let's say if this is the stem, this is the node, and from the node is going to arise our stalk of the flower, which is known as pedicel. This one is known as pedicel. The stalk of the flower is known as pedicel, and this portion is called thalamus. This portion is known as thalamus. where all the floral structures will be present, also known as receptacle. Thalamus is also known as receptacle. Now guys, what happens? Now, there are certain structures. See, this is the first structure that I'm going to discuss. This is the pistil or the carpel. This is the pistil or the carpel. Yes or no? Clear? Now, this is a very basic structure that we are already known to since childhood, right? This is a typical structure of the flower. And this one is known as stamen. Now, actually, these are the two main reproductive parts of the flower. These are the two main reproductive parts of the flower. Please let me know that these points are clear. These are the reproductive parts of flower. Why? Because they actually, and they directly participate in sexual reproduction. Are these points clear? Are these points clear? Right? Okay. Now see, this is the female part and this stamen, it already has men word. This is the male part of the flower. Please let me know that these points are clear. Now, other than these, there are some other structures which makes the flower more colorful and attractive. These structures are called petals. These structures are known as petals. And to protect the flower in the bud stage, we have these leaf-like structures known as sepals. Are we all very thorough with these terms? This is very basic. I've not even started yet. Now, taking it a step ahead. Now we know that collective term used for pistil and carpel. We have read it in the chapter of morphology, guys. This is known as gynoecium. This is known as gynoecium. The collective term for all the carpels is gynoecium. The collective term for all the stamens is known as androecium. Andro word is for males, androecium. Then the collective term used for all the sepals is calyx, while the collective terms for all the petals is corolla. Are these points clear? And now, sepals and petals do not directly get involved in sexual reproduction. They are going to become an aid for sexual reproduction. They are going to help in that. So that since they are not directly involved, we call these structures as accessory structures. We call them as accessory parts of the flower because they are not directly involved. Are these points clear? Are these points clear to all? Please let me know. Yes, guys, please let me know. Buffering. All right, guys. All right. Let me check. Why is the lag? Lag हो रहा है. 4 चाहिए था मैंने. नहीं आ रही. All right, guys. All right. I think it is fine now. Is that clear? All right. I think now it is absolutely fine. Okay. Now, are these points clear? So now we know that petals and sepals are the accessory parts, while pistil and stamen, they are the reproductive parts. Now, since we are going to talk about the sexual reproduction and the process of it, we have to mainly deal with these two structures, that is gynoecium and androecium. Is that clear? Now, starting with the first male part. So today's session will be basically about the male part. So please understand. Now see, the male reproductive part of the flower. Male reproductive part of the flower, which is nothing but stamen, right? Now guys, we have just read that basically all these floral structures, see, I've told you leaves have modified into floral appendages. So for leaf, we use the term fill. And since it is a male, it is a leaf which is going to bear, this is a modified leaf. See, stamen, it is a modified leaf that bears male spores. Therefore, Therefore, can I call stamen as microsporophil? Please tell me if you are clear with the point. I can call stamen as microsporophil because it is a modified leaf that bears spores, and that is why micro word is only for males and sporophil means leaf that bears spores. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. So, male reproductive part is stamen, and what is stamen? What does stamen represent? Stamen represents that leaf which bears the male spores. Hence, we call it microsporophil. Are these points clear to all? All right. Now, let's see the structure. Let's see the structure of a stamen. So, we know this is the thalamus, and on the thalamus, guys, are present these stamens. I'm going to draw just one stamen. This is one stamen. Now this part is known as filament. This stalk is known as filament, while this portion is known as anther. This portion is known as anther. Please tell me these points are clear to you all. Please tell me these points are clear to you all. So this is filament and this is anther. These are the two parts of the stamen. These are the two parts of stamen. Two parts. One is anther, second is filament. So filament is what? Filament is a stalk. Now this portion is this region which is towards the thalamus. This is the proximal end. This is the proximal end of filament, while this end is the distal end. Now there is this line in NCERT that the proximal end of filament is attached to the thalamus, while the distal end is attached to the anther. Do we understand? So first of all, you should be very clear about these two terms, proximal and distal. So proximal means near. So the region which is nearer to the main plant body is the proximal part, and the region which is away from the main plant body or the central axis, this is called the distal part. Please tell me if these points are clear. Perfect. All right, so this is the stalk. Now talking about this anther. So there are certain characteristics that you should be very clear about anther. We are going to use certain terms and I'm going to clarify these terms one by one. So first of all, your anther is bilobed. See, these features are typical. For a typical anther, these properties will be true. So a typical anther is bilobed. It is diathecous. It is tetragonal. It is tetragonal, and a typical anther is tetrasporangiate. Tetrasporangiate. So now I have already introduced you to 4 different new terms. So a typical anther is bilobed, it is diathecous, it is tetragonal, and it is tetrasporangiate. Now let's understand, let's decode these terms one by one. So talking about the anther. See, first of all, I'm going to make it large in size. This is the anther. Okay. Do you see that our anther has 2 lobes? So it is 1 and this is the second lobe. Do we see that? Yes. So since it has 2 lobes, there are 2 lobes. A typical anther has 2 lobes. So that is why it is known as bilobed. Please tell me if this point is clear. Please tell me if this point is clear. See, we are only going to talk about the typical features because exceptional cases are always present. Exceptional cases are always present. Is this clear? There are certain angiosperms which will have only 1 lobe. So I am using the word typical again and again. Right? Now, do you see that there are 4 different corners? 1, 2, 3, 4. Do we see the 4 corners? Yes or no? So since it has 4 corners, we use the term tetragonal. Therefore, it is tetragonal. Please tell me that this point is also clear to you. So a typical anther has 2 lobes and it has 4 corners. Please tell me these points are super clear. Now, other than this, each lobe will have 2 Each lobe will have 2 sporangium, microsporangium, or you can call it later on when it will be filled with pollen sacs, we are going to call it as pollen sacs. So since each lobe has 2 microsporangium, therefore we use the term it is dithecous. It has 2 theca, so we call it dithecous. Please tell me these points are clear. There are 2 theca. Please tell me this point is super clear. Is that clear? So in total, guys, in both the 2 lobes, in total, how many pollen sacs will be present? It will have 4 pollen sacs. 4 pollen sacs, or we can say 4 microsporangium. Therefore, we call it tetrasporangiate. We call it tetrasporangiate. Yes, please. Tetra means 4. Sporangium word comes from microsporangium. It has tetrasporangiate condition. Please tell me that these 4 conditions, these 4 characteristics of a typical anther are clear to you. It has 2 lobes, bilobed, it has 4 corners, tetragonal, and it has 4 pollen sacs, tetrasporangiate, and since there are 2 theca, we call it dithecous. Yes, guys, please let me know. All these points are super clear to you all. Yes, please. Is that clear? Shahzeb, I am on Unacademy NEET English channel, and this is a demo class for batch Phoenix 2.0, which will be preparing you for NEET 2027 if you are a class 12 or a dropper student, and I'll be taking those batches. Please tell me all these points are clear. Please tell me these points are clear. Okay, I'll change the color. Yes, microsporangium contains pollen grains. That is why we call it microsporangium. Pollen grains. Is that clear? Okay. Are these points clear to you all? So do you know why male part stamen is called microsporophyll? Because this leaf bears these pollen sacs, microsporangium, and now these microsporangium coming here. I'll make the structure again. See, now what's going to happen? See, this is right, and there are 4 pollen sacs. Now these pollen sacs, guys, they are filled with these cells, special cells. Now these cells are known as, what are these cells known as? These cells are microspore mother cells. These are known as microspore mother cells, or you can call it pollen mother cells or pollen mother cells. Please tell me these points are clear. So each cell will be a pollen mother cell. But if I have to name the tissue, then what is the tissue known as? The tissue is known as sporogenous tissue. NEET 2026 has asked this question, sporogenous tissue. So there was this question in NEET 2026, arrange them in the correct sequence. Now see, first of all, we have sporogenous tissue. What does sporogenous tissue contain? It contains microspore mother cells, and mother cell always gives rise to a particular cell. So what will microspore mother cell give rise to? It will give rise to microspores. It will give rise to microspores, which are commonly known by the name of pollen grains. Please tell me that these points are clear. Please tell me these points are clear. So that is why these microsporangium will be known as pollen sacs. They will be known as pollen sacs. Are these points clear to you all? Please tell me, guys. Is that clear? Is that clear? Yes. First of all, it will form. So from one microspore mother cell, 4 microspores will be formed. But how? We'll just start with that process as well. Please tell me these points are clear to you all. Yes or no? Yes or no? Please tell me. Quickly give me a thumbs up that all these points are clear. Please give me a thumbs up. All these points are clear. Yes, please. Quick, quick, quick. Super. All right. Now moving on to the details of. Now see, we have talked about the main thing, right? But what about this area? This area. This represents the wall of the anther. This represents the wall of the anther, and we have to talk about this wall of the anther also because it is going to play important role around this sporogenous tissue. So now I'm going to draw a transverse section, guys. I'm going to draw a transverse section of anther, which is little butterfly like, and in this structure you can easily see that there are 4 corners. Quickly give me the name. What is the condition known as when it has 4 corners? Quickly let me know in the chat. We have just read. So there are 4 corners, 1, 2, 3, 4, and in each corner guys, we see a pollen sac or a microsporangium. Please tell me. And this region is known as connective. This is known as connective, where it is connected to the filament, and vascular tissue will also be entering through this. Yes, please. Quickly let me know. What is the term used for? What is the term used for? Ma'am, which team are you in? So guys, my team is a complete English batch team, and this is my team. Yes, this is my team, guys. So together we all are going to help you prepare, and this will be a complete English batch, while there are other English batches also. So whatever batch you take, you can always attend the classes. Yes. Is this point clear? Is this point clear? Yes, I'm going to continue the entire year, and we are going to complete our syllabus, both botany and zoology. Is that clear? No, not tetrasporangiate. Guys, Monica, I am asking what is the term used for the anther which has 4 corners? 4 corners, I am saying. The term used for 4 corners is tetragonal. Let's have a quick look. See? 4 corners, 1, 2, 3, 4. It is tetragonal. 4 pollen sacs means tetrasporangiate. So please get these terms right. Now, this is a pollen sac, or it will be later on known as a pollen sac when it is filled with pollen grains. For now, we'll call it microsporangium. Yes or no? Okay. And also we know that this one is all filled with the tissue. I've just told you it is a sporogenous tissue, and each cell will be known as microspore mother cell or pollen mother cell. Please tell me these points are clear. Please tell me these points are clear. Yes, please. Is that clear? Everyone? Super? All right. Now talking about this wall. First of all, guys, the outermost layer of the anther, this outermost layer is known as epidermis. Just like our skin, this is also known as epidermis. Can I call it epidermis? Please tell me. Then inner to epidermis, guys, inner to epidermis, here there will be a single layer again, and now this layer is known as endothecium. What are we going to call it? Endothecium. Yes or no? Please tell me. I'm going to rub this so that we can just concentrate on this portion for now. I've told you already, this one is connective. All right, are the 2 layers clear? The outer one is epidermis, inner one is endothecium, and now still moving inwards, so there is this 3rd layer, and it is made up of 2-3 layers. These layers, guys, are known as middle layers. What do we call them? Middle layers. Yes? Clear? So we have a single layer of epidermis, we have a single layer of endothecium, while middle layers are 2 to 3 in number. Middle layers. Clear? And then next to the middle layers, guys, we have this. these elongated cells, we have these elongated cells, and these elongated cells or the innermost layer is known as tapetum. Please tell me this is clear to you all. First of all, you should be just clear about the arrangement. So if the question comes and it is already a PYQ, arrange the 4 layers in the correct sequence from outside to inside. Can you go outside to inside? So this is outside and this is inside. So outside to inside. Yes, please. Can we mark that? Can we mark that outside to inside, guys? So the outermost being, this is the outermost, outer to inside. Yes or no? Outer one being epidermis, next to it is endothecium, then middle layers, and then tapetum. Please tell me that these points are clear to all of you. Yes. So these are the layers present in the wall of Anther. Now, second thing is to understand the function of each layer. Now, epidermis, endothecium and middle layers, since they are the outer layers, guys, these are the outer layers, they are going to be protective in nature. They are going to be protective in nature. See, all these, they are protective. So they provide protection. All right. So they are protective. Please tell me. The first 3 layers, the outer 3 layers are protective, while tapetum is nutritive. Nutritive. Is that clear? Not only protection, see, once the pollen grains are formed, this anther must release the pollen grains. It is going to burst open. It is going to dehiscize. It is going to spread the pollen grains. How are the pollen grains going to spread? So basically, the epidermis and endothecium, they are going to help exactly in dehiscence. They are also going to help in dehiscence. So second function of these outer layers is dehiscence. It is dehiscence. Dehiscence means spread. So the outer 3 layers play 2 functions. One is protection and second is dehiscence. Dehiscence means spread of the pollen grains. Are these points clear? Now, I already told you this is single layered, this is single layered, there are 2 in number, and this is also single layered. Now, tapetum is not just nutritive. This is also going to, see, first of all, the tapetum has 2 to 3 nuclei. Remember, it is nutritive. Why nutritive? It is binucleated, at least binucleated to multinucleated. It can have 2 or more than 2 nucleus. Now, what is the significance of nucleus here and its relationship with providing nutrition? Now, nucleus is the hub for protein synthesis. Yes, nucleus has DNA. How this DNA is formed, we'll study it all in the chapter of molecular basis. Now, tapetum has at least 2 nucleus, 2 to more nucleus. More the number of nuclei, more is the protein synthesis, more and better nutrition it can provide. So that is why tapetum is binucleated, to provide proper and more nutrition to the developing nutritive to the developing microspores. Please tell me these points are clear. Please tell me these points are clear. Yes or no? And it is binucleated. Now, so this is the first function. Other than providing nutrition, guys, it also produces sporopollenin. Now, this is a special pollenin. This is a special chemical which is present in the, which is going to be present, which is going to be present in the outer wall of pollen grains. This is such a miracle chemical which is not destroyed by any microbe or any acid or any base. Please tell me all these points are clear. Please tell me all these points are clear. Is this clear? Is this point clear? Chalo. Is that point clear to all? So guys, nutritive, binucleated, and it produces sporopollenin Clear to all? Now, where and what is the significance of sporopollenin is, we'll be discussing it one by one. Now, please tell me if these points are clear to you. Please tell me. Also, now tapetum is going to produce callase. What is the function of callase? We'll be discussing it. It is also going to produce callose. Now these two will be discussed once I start with the process of microsporogenesis. Please tell me. Hanji, are these points clear? Are these points clear to all? Right? Okay. Now moving on to the process. So we have seen the structure of Anther. Okay, we have seen the layers present in the wall, and we have also seen the type of tissue present in the microsporangium. Now what happens to each and every pollen mother cell? What happens to every cell? How are microspores formed? How are pollen grains formed? Now see here. Let's see. Yes, guys. Okay. Now, first of all, I'm going to talk about one microspore mother cell, or you can call it pollen mother cell. So first of all, this pollen mother cell is diploid. Remember that. So far, all the structures, be it root, be it stem, be it leaf, be it any other part of the flower, till microspore mother cell, all cells are 2n. Clear? Okay. Now, this microspore mother cell, guys, is going to undergo first division. It is going to undergo first division. And it is going to undergo first division, which is meiosis, and form 4 daughter cells. So we know as a result of meiosis, how many daughter cells are formed? 4. So it is going to form 4 cells. It is going to form 4 cells, and these all 4 cells are together. Now these cells are known as microspores, but because they are held together in the packet of 4, we call this packet as microspore tetrad. What are we going to call it as? We are going to call it as microspore tetrad. Now why are they held together? Here comes calyx. So guys, around the microspore mother cell, there is a coating of callose present. There is a coating which is known as callose. Around every microspore mother cell is present callose, and we have just read who produces callose. Callose is produced by your tapetum. Is that clear? So around every microspore mother cell we have a callose. Now when it undergoes meiosis, it is still surrounded by callose, and that is why all the 4 cells are staying together in the form of a tetrad. Is this point clear? Now what will happen? This tetrad will disperse, because here comes another enzyme. Do you see? Callase. If there is ase after any name, it is an enzyme. Now callase comes in and it dissolves the callose. Callase dissolves the callose, right? Since callose is dissolved, now what will happen? All the microspores will be separated. Is that clear? All the microspores are separated. Please tell me, how many microspores are present now? 4. Anyone? Now see guys, it is completely a matter of observation now, and little bit using some extra knowledge. See, let me know in the chat, from one microspore mother cell, how many tetrads were formed? Yes. Please tell me. Guys. How many tetrads are formed from one microspore mother cell? Quickly. Now this is completely observation based. Yes, please. How many tetrads are present? Quickly let me know. Yes, please. 4, ma'am. Povish. 4. Anyone else who wants to answer? How many tetrads are present? I'm not asking for microspore, I'm asking for tetrads. Tetrad is a packet of 4. How many packets are formed? If I have to ask again, how many tetrads? See, tetrad is equivalent to 4 microspores, or 4 microspore equivalent to 1 tetrad. So from one microspore mother cell, how many tetrads are formed? One tetrad is formed. How many microspores are present? 4 microspores are present. Please tell me if this point is clear to you all. 5. Already in flying color situation, huh? 5, 2, random, anything, huh? No, guys. So from one microspore mother cell, how many packets of 4 tetrads are formed? 1 tetrad. How many microspores are present? There are 4 microspores, but they are held together, so we call it a tetrad. But then it is called a microspore. Now this was NEET 2026. NEET 2026. What is the correct sequence? So first of all, we have sporogenous tissue. So in sporogenous tissue, we have microspore or pollen mother cells. After pollen mother cells, what are formed? Microspore tetrad. And from tetrad, the 4 microspores separate, and this is the correct sequence. This is NEET 2026. Please let me know that this point is clear. And guys, this completes our process of microsporogenesis. Because microspores are formed, this process will be known as microsporogenesis. This is known as microsporogenesis. Formation of microspore from mother cell. This is microsporogenesis. Please tell me if this point is clear. Yes or no? Also, let me know in the chat what type of cell division has taken place in microsporogenesis. Yes, guys. What type of cell division which took place? Cell division. Sorry. What type of cell division has taken place during microsporogenesis? Cell division involved. Quickly let me know. What is the cell division involved? Quickly. What is the cell division involved? Meiosis. Absolutely. The type of cell division involved is meiosis. Right? Next question. How many meiosis takes place during the sporogenesis, microsporogenesis? Yes, please. It is reductional. Yes, that is why. I'll come on to that also. How many meiosis have taken place? Yes, please. How many meiosis have taken place? How many? There is just one meiosis that has taken place. Right? Now, if I said the ploidy of microspore mother cell is 2n, what is the ploidy of each cell in the microspore tetrad, or the ploidy of each microspore? Quickly let me know. What is the ploidy of each microspore formed as a result of meiosis? Yes, please. How many? Naman, I'm already married. So meiosis 1 and 2 both, yes. And how many meiosis will take place? One meiosis will take place. I am asking, if the ploidy of microspore mother cell is 2n, what is the ploidy of microspores? Haploid. Absolutely. Absolutely. This is n. So it will have half the number of chromosomes. So this part is known as microsporogenesis. Please tell me if this point is clear. So basically, your pollen grains have been formed. Pollen grains have been formed. Is that clear? Now, the next step. Now, please understand. Now, this microspore mother cell which is formed, sorry, this is microspore. This is one microspore. And it is N. Also, let me mention here, all the 4 microspores are functional. All 4 are functional here. So each microspore is going to undergo the following changes. Now what will happen? This microspore will undergo mitosis. It will undergo mitosis in such a way that 2 cells will be formed. two cells will be formed. Now this is also a microspore. Please see. This is also a microspore. The difference being this is one-celled microspore and this one is two-celled microspore, but still a microspore. Now another point to be noticed here. So since there are two different sized cells formed here, the larger cell is known as the vegetative cell. See, in biology, vegetative word is for, I'm not talking about vegetable, I'm talking about vegetative. Vegetative means non-reproductive. So this cell will not be involved in further reproduction, while the smaller cell is known as generative cell. The smaller cell is known as generative cell. So we have one generative cell and one vegetative cell. But do you see, out of mitosis, there are two different sized cells formed, which is not usually a case. Now, point to be noticed here is, just a second. Yeah, that this mitosis is going to form asymmetric spindle. I hope you remember that there is spindle formation during cell division, yes or no? Now, because there is asymmetric spindle formation, that is why it results in the formation of one large cell and one small cell. Is that clear? This will not further undergo any division or any this will be involved in, this will form a pollen tube or a pollen nucleus. This nucleus will be known as pollen. So nucleus will be known as a tube nucleus. Yes, this will be known as a tube nucleus. Please tell me these points are clear to you all. So vegetative cell is large and it will help in the pollen tube formation, and the nucleus is known as the tube nucleus, while this one is a small cell and will form gametes. How? We'll see. Now what will happen? The generative cell, only the generative cell will undergo another mitosis and it will form, see. So this is the vegetative cell we have just talked about, right? Now this one will undergo second mitosis and there will be two nuclei formed, and these two are the male gametes. These are the two male gametes. So basically generative means it is going to give rise to generative cell, it gives rise to male gametes. Please tell me these points are clear to all of you. And guys, because here the male gametes have been formed from one microspore, that is why this process will be known as microgametogenesis. Formation of gamete from the microspore. Is that point clear? Is that point clear? Please tell me. So formation of gamete from the microspore is microgametogenesis. And what type of cell division is involved here? Cell division involved? Mitosis. And how many mitoses? Two mitoses. So see here, if I say the story of one pollen mother cell, it is going to form four. It is going to form one microspore tetrad, right? Now microspore tetrad will have four microspores. This process is called microsporogenesis, right? We know this is 2n, and then each cell here will be n. And what is the cell division involved? This is meiosis. Next, from each microspore formed here, one microspore, it will form a two-celled microspore and then a three-celled microspore. Three-celled microspore. How many or which divisions are involved here? It is mitosis. First mitosis, second mitosis. Please tell me these points are clear to you all. And this process will be known as microgametogenesis. Yes, please. Please tell me all these points are clear to you all. Please tell me all these points are clear to you all. So there is this question for all of you to answer. Yes, please. You have to answer, guys. You have to answer, guys. You have to answer how many total meiosis and mitosis take place in one pollen mother cell. Number of, you have to answer this question in the chat. Number of cell divisions involved in one pollen mother cell to form male gametes. Let me know the answer in the chat. Let me know the answer in the chat. It is right in front of you. And those of you who will watch this later on, you have to answer this question in the comment section. If you can answer this question, we are on the right path, guys. We are on the right track. We are understanding every concept. And we are moving ahead towards winning NEET 2027. And if you want to study in similar way, where we understand, where we decode each and every term, we understand the whole process in detail, where we are ready for all types of questions from this topic. So don't forget to join my batch, that is Phoenix 2.0 for NEET 2027 by the Team Avengers. Me and my team, we are with you for the entire year and help you succeed. Let me know in the comment section, guys, and don't forget to enroll quickly because the batch is starting on 20th May, and as said, the fees will be 4,699, only valid till May 10th, only if you use this code TRLIVE. So don't forget to use this code and enroll quickly so that we study together. You'll get a proper schedule for every day. You'll know what is going to happen every day. You'll get proper notes, both in plus as well as in iconic batches. So you'll get a personal mentorship, guys. Is that clear? So get ready with us for NEET 2027. Let's learn from our previous mistakes. Let's work wiser, work harder, and let's crack NEET 2027. So let me know in the comment section, how did you like the lecture? I'll see you soon in the lectures in Phoenix 2.0 batch. Thank you so much. Good night and let's crack it. Bye.